important, but this would be good too. Hopefully you see sort of the same tree. The difference is that this one splits ctenophores and cnidarians together. It shows sponges as being paraphyletic. It brings the acials out of platyhelminthes and puts them into a sister relationship with the other bilaterally symmetrical animals. And of course, the placozoans would go here. They were historically thought to be cnidarians, but they're brought out now, and they're probably the sister group to all of the eumetazoa. Yeah. So I just showed you this. Sponge, sea anemone, jellyfish, ctenophore, placozoan, and acial, right? So here we go. So the peripheral, which is probably not monophyletic and probably going to be broken up into at least a couple different groups like Calcarea and Cilicia, right? Sponges are all sessile. What does that word mean, do you know? Yeah, well, not really that they completely don't move, but basically that they attach to the bottom somewhere and live their life attached to the bottom. They don't get up and walk around, right? There could be moving parts, like they have little flagella and stuff that are moving, but they, as the whole sponge is concerned, it sits attached on the bottom somewhere. You'll see when we go to moat, I'm going to ask you to use words like benthic, right, versus like planktonic, right? So the word planktonic means what? Means that you float, like zooplankton, little like larvae and things like that that float. Phytoplankton are little algae and stuff that float, right? Benthic means you're on the bottom. So, for example, you could say that a sponge is benthic. It is, right? It's living on the bottom, and it's also sessile because it's attached on the bottom. It doesn't get up and wander around. But how about something like a crayfish? Crayfish is benthic too, right? Crayfish don't float, and they're not like up swimming around like this. I mean, where's a crayfish? If you go to a lake, where's the crayfish? Do you know? He's down on the bottom. He's down running around in the mud and stuff. But you wouldn't really say that a crayfish is sessile because he doesn't just sit in one spot all the time. He gets up and walks around. So it's important to be able to distinguish between these terms, benthic, which just means you're on the bottom, and the word sessile, which means you basically are attached to one spot. You don't get up and wander around. Sponges are sessile. They don't wander around. They don't have muscles, remember. They don't have nerves. They have no true tissues. So mobility would be seriously limited by that alone. They filter feed. The filter feeding occurs at the level of single cells. That's why you can almost think of this as a bunch of individual cells living their little lives within a colony rather than as a true multicellular organism. There is no gut. They don't undergo normal gastrulation during development or anything. There is no centralized digestive tract, right? You realize, for example, your skin cells are not out here like eating a little bit, every skin cell eating a little bit, right? You have a centralized digestive system that is responsible for breaking down food, and then the nutrients are passed to your bloodstream, and then they are carried throughout your body to all the different cells. But the other cells that are not an immediate part of the digestive tract are not focused on ingesting food particles and digesting them, right? That's not the case in a sponge. In a sponge, every single cell is responsible for taking in food and feeding itself, right? So it's a very simple organization compared to what you typically think of in an animal. They're all aquatic, which is probably not a big surprise because if you're a filter feeder, it's a little hard to filter particles of food out of the air, right? If a sponge was sitting on the forest floor, it would have a hard time getting food to come to it, right? Air is not going to really carry food the way that water would. So they're all aquatic. There's none that live on land. They don't have any true tissues. There's no gastrulation. They basically have two different cell types. I already showed you those quinocytes, those little collar cells that look like almost little individual quinoflagellates, right? And then they also can have the second cell type called amoebocytes. I'm not sure every sponge has those, but there's some do. They have these skeletal elements called spicules. They can be made of either calcium carbonate or silica. Silica just being glass, basically. That appears to be phylogenetically important, so that the sponges that make silica spicules are not really probably related to the ones that make calcium carbonate spicules. They have an extracellular protein, these protein fibers, these collagen fibers that are elastic. It's called spongin. This is why when you take a sponge and you squeeze it like this, it doesn't stay squeezed. It goes like this and pops back, right? That's because of these collagen fibers. What a sponge is basically it's the skeleton and the spongin of an old sponge, right? The cells are all gone, but the protein fibers and the skeletal elements are still there, right? They still do that spongy thing. They typically shed gametes into the water. They're obviously not going to get up and copulate or anything. Right? They just sort of let the sperm and the egg go, and fertilization happens externally out in the water somehow. My understanding is a majority are hermaphroditic, but there are a few sponges that are separate sexes, so like boy sponges and girl sponges. But most of them shed both eggs and sperm into the water. And the larval stage for a sponge, the typical larva is called a parenchyma larva. Remember on that, in the lab manual, how you're trying to fill out those characteristics for each group? I think it asks you like about larval type. This would be maybe what you could write for it.
Is a sponge, did you say sponges were unicellular?
Okay, well, quinoflagellates can be considered unicellular. So if you include quinoflagellates as being unicellular sponges, and you can argue that they're, most people wouldn't do that. Most people would say they're quinoflagellates. But in essence, they're kind of like unicellular sponges. Otherwise, the things that we refer to as sponges up here are all multicellular, depending on how you define multicellularity.
When they come together, they're, but an individual sponge
My argument here is I said, what would you consider sponges if you didn't consider them to be true animals? And my argument was that you could consider them to be a colony of cells. So if you think of something like Volvox, remember when we have Volvox, which is basically a bunch of little independent Chlamydomonas-like cells that are all held together in an extracellular matrix, so that they kind of live together, but there's no differentiation between them. All the cells are basically exactly the same, and each little cell is basically fending for itself, other than the fact that they all sort of live together. You could make an argument that that's really a bunch of individual cells that are living together like they're in a little colony, but that it's not true multicellularity. The true multicellularity, you really have to have almost a division of labor where certain cells do one job and other cells do another job, such that each little individual cell would not be able to persist on its own. Their fates are all very tightly tied together. So let me show you that kind of as an example when we do this here. So we have the quinoflagellates, right, but we're going to focus now on sort of these sponges. I'm not sure I stuck that slide in, because I guess I thought you didn't have it. This shows you a cartoon of a sponge. Remember, again, these are very difficult to see in real life. They're very small. So if you see this little flagellum beat, here the food particles are being brought in, and you see the cell just undergoes phagocytosis in order to bring food particles into a food vacuole. The amoebocytes, basically food vacuoles or food particles are transferred to the amoebocytes, all just kind of by phagocytosis and by beating this little flagellum to bring them in. The beating of the flagellae, so they are working together in the sense that water is drawn in through the pores and up through this osculum. So there's some working here, but at the same time you can see these are individual little quinocyte cells, these are individual little amoebocyte cells. Here the skeletal elements are the spicules, which are just extracellular, right? They're secreted to the outside. You have these little pores that water's brought through. There's no nerves, there's no muscles, there's no real organization of anything. There's certainly no gastrovascular cavity. This is really, really simple. If you came in here and you like shot these three cells and they died, I don't think that affects anybody else, does it? Right? If I went in and like shot 3 of your brain cells, that's probably going to affect something else, right? But these are just 3 little cells that are just like this cell or just like these 3 little cells. It's almost like each one of these is its own little individual, and they just all happen to be living together. I wouldn't really think of like one of your skin cells or one of your brain cells as being an individual, right? It kind of is not worth anything away from you, right? But each one of these little guys, it's feeding itself, it's doing what it does, moving its little flagellum the way that it does. I mean, there's really a lot of sort of almost independence of them, such that there's not a whole lot of differentiation or different cell types. There is a little bit, right? You have these amoebocytes versus quinocytes and stuff, so there's a little bit of cell differentiation. But yeah, so the question is this multicellular? I don't know. It's going to be touted as multicellularity. Most books are going to sort of argue that these are multicellular animals. That's why, so for example, Whitaker would have considered a quinoflagellate not an animal. He would have thought this was an animal because he would have argued that this is multicellular. I think you could make an argument that this is not true multicellularity, that this is more colonial arrangement. It's a matter of interpretation, right? I mean, it is what it is. How you want to interpret it, though, is kind of up to you. Most biologists would probably consider this to be very, very simple multicellularity.
Can something that's unicellular do meiosis?
Yeah.
They can.
Yeah. Right, so Chlamydomonas does.
Huh?
Chlamydomonas does, right? Chlamydomonas, basically, there's 2 haploid cells. They fuse together to make a diploid zygote. It does meiosis to make 4 haploids.
Okay.
Yeah, there's nothing that prohibits you from doing meiosis or mitosis by being unicellular.
Okay.
Yeah, are we all good with this? So exactly how you want to interpret it, that's what it is. But those are sponges. That's all I'm going to do with sponges. You can learn them all as phylum, peripherum. We'll just leave it at that. So now we come to Cnidaria. This is going to be, we're going to treat this a little bit more. The niddo refers to a nettle. Y'all know what a nettle is? A plant that if you touch it, stings, right? It refers to those stingers that they have that you all looked at, right? Called nematocysts. Remember you watched the little hydrozoid fire in nematocysts and you were looking for those 4 types of nematocysts? I'm going to get down to this in a second, but that is a great character to define this group. As far as I know, here's your mission to go out and prove me wrong. As far as I know, every cnidarian has a nematocyst, every one. And no non-cnidarian has a nematocyst, including ctenophores. Everybody's like, oh, they're jellies, you know, they must be like jellyfish. They don't have nematocysts. This is a great synapomorphy for this phylum. This is the defining character. It's extremely distinctive, and it's found only in this group of animals. I mean, it's really kind of, it'd be shocking if this was not a good group of animals. It's not a real group. They have radial symmetry. What does that mean again?
A circle? Sure.
So basically their body is circular. If you kind of want to think of it that way, it means that there are multiple different planes that you could pass through a central axis and end up with two mirror image halves. They don't have a right and a left. You'll see that we tend to refrain from talking about like anterior, posterior in these guys. Instead, we tend to say the oral side and the aboral side. So the side the mouth / anus is on, and the other side, basically. We tend not to talk about them having anterior, posterior, because that kind of implies that you have what? Head and a tail and a left and a right. These guys don't really have that, right? They have a blind gut, meaning what I just said, they have a mouth / anus. They eat and defecate through the same hole. They have radial cleavage. What does that mean? Oh, I'm afraid you're going to say, what does that mean?
You
What
can't
I'm
break
really
it off.
hoping is that you realize that this has absolutely nothing to do with this, right?
You can't
This
break
is
it.
talking about overall body symmetry. This is talking about what?
The cell can't be broken off.
Yeah, this has to do with when cell fates are established. The other way to say this would be they have indeterminate cleavage, meaning that cell fates are not established at the first division of the zygote, right? So you could have identical twins here. They have basically what we have, right? Which is probably the ancestral condition. All of the determinate cleavage patterns that you see mostly in protostome animals are going to be probably derived, right? They have different life stages that are characterized by these general body forms called a medusa and a polyp. You looked at these in the lab, right? Basically, both of them are a bag with tentacles. Basically, a polyp is sessile. You all know what that means now, right? A bag with the tentacles facing up. A medusa is pelagic. It swims. Hopefully those look similar to you, right? Most cnidarians, or a very large number of cnidarians, actually are juvenile polyps, and then they bud off and become medusae as the adult stage. As far as I know, all jellyfish do that. Obelia, that little colonial hydrozoan you look at, does this. There were prepared slides actually of both stages. This was the one you were really supposed to look at, but we had prepared slides of the little medusae up there too. Nematocysts, I already mentioned, are those stinging cells, right, that are the hallmark feature of cnidarians. Cnidarians are touted as being diploblastic, meaning that there's only two germ layers, just endoderm and ectoderm, with this extracellular matrix between the two germ layers called mesoglia. But as I mentioned, sometimes there's cells inside of that mesoglia, so some people have argued that some of these are actually triploblastic. They have two mesoderm. If you have cells in there, then that would argue that it's a third germ layer. So that's maybe a little bit debated. But as far as you guys are concerned, we're going to just say they're diploblastic, right? Because that's the way they're typically taught. The arrangement of muscles in cnidarians are circular and longitudinal muscles because their bodies are basically round, right? So they don't have jointed skeletons or something with little flexors and extensors, right? You have circular muscles and longitudinal muscles that work antagonistically. So your little hydra, when he got long and skinny, how did he do that? The circular muscles are contracted, the longitudinal muscles are relaxed. When he gets short and fat, how did he do that? He contracts the longitudinal muscles and relaxes the circular muscles, right, to get short and fat. And you might remember I said in the case of the medusae, there actually are only circular muscles. So there, how does that work? When the circular muscles contract, it forces water down from the belt, push him forward, right? But then how does he go back again? Do you remember? It's like a spring, right? The mesoglia actually acts to store potential energy, and then when he just relaxes, it pushes it back, right? It's like storing energy in a spring. So it's an example of something that's serving as an antagonist to a muscle that's not a muscle. You're looking for a larval stage for cnidarians, a planula larva. Hydra, remember, is a weirdo. Remember Hydra, the little poster child that you had for a hydrozoan? Hydra's a weirdo in that there's no planula larva. I'll show you a funny little story with that in a second. There's no medusa stage in Hydra, which is a little bit weird. Hydra can be hermaphroditic. That's a little bit weird. Most cnidarians are separate sexes. And Hydra, remember, has 4 nematocyst types, which that's weird too. So here we go with showing you an example of a hydrozoan, a cnidarian, the one that you're going to know. And there's all these things about it that make it a little weird, but it's the one that everybody knows as an example. So usually they have separate sexes. They're boys and girls, right? But they also are very good at this, with the exception of Hydra. Although there are some separate sexed hydras, but a lot of the species are hermaphroditic. They have this clonal growth called budding. Did you all see this in Hydra? Where Hydra makes like a little baby Hydra right here and it just buds off. It's a little clone of it. And obviously like corals do this all the time. So a lot of the coral polyps are all just clonal individuals. They're all genetically the same, all the different little polyps that make up the coral. And so especially in the polyp stage, this budding is very common. This shows you a polyp and the medusa. You'll notice there's no red on here because there's no mesoderm, right? Instead, you have just two germ layers. You have the ectoderm, making up the epidermis here. You have the endoderm, which is shown as the lining of the gastrovascular cavity. There's only a single opening to the gastrovascular cavity. It's a bag of tentacles. And then you have this mesoglia, which is the gelatinous extracellular matrix that's between the endoderm and the ectoderm. You know what I've done on exams? I go copy, paste, delete all the names, have you fill them in. What a disaster that was. I had people put Salome, mesoderm, all kinds of crazy stuff, right? These are really, really simple animals. There is no Salome here, or anything that even approaches a Salome. You've got just basically these two tissue layers with an extracellular matrix between them. You've got a gastrovascular cavity with a single opening, right? This shows you the nematocysts in Hydra. Here's the tentacle. Here's a little cnidoblasts, or cnidocytes is the word I usually use for them. The little trigger, right? Here you've got the thread, the stinging thread with the venom in it, all coiled in, right? And then, of course, it discharges very quickly, and that thread penetrates the prey or whatever it's trying to defend itself from and injects the venom. And the variability of the venom, some of the most venomous animals on Earth are cnidarians, right? Some of these box jellies and stuff, which could kill you in like a second. And then others, like that little Cassiopeia, you touch it, you don't even feel it because its nematocysts don't penetrate your skin. They're not very potent, at least not to you. They're not. This shows you the Hydra one. Remember, Hydra again is weird because it has these 4 different kinds. Did you guys see all these different kinds? Like on the prepared site, you could definitely see these ones with a little pigtail like that. And I'm hoping y'all saw these. Did y'all see these with the barbs on them? I saw them. I think it both ran pretty well. When you were looking at the real Hydra, here you see it look kind of like a little horseshoe. You can't see very well the little barbs. And these show you the kind of unfired ones, right? So this is kind of like before you squash him, here's his tentacle, right? And there's kind of after you squash him. Y'all saw that, right? You realize I always put the nematocysts on the lab practice, but it's really easy. Okay, so here's my funny story. So remember I was talking about the larval stage that most cnidarians go through. It's called a planula larva. And you'll notice it's this little ellipsoid thing with cilia and it swims around. And you remember I studied mushrooms, right? So first semester I was here, we were doing this, and we had some old Hydra, and a student was looking at them, and I looked in there, and just under the dissecting scope, and here in a little watch glass, there's a little Hydra, and there's all of these little things that are kind of elliptical, and they have like all these little cilia, and they're just running up and down the little Hydra all over his little tentacles. And they look like little puppy dogs, you know, like if you came and you had like 10 little puppy dogs, and they were all excited to see you, and they come up and they just rub up and down you and lick all over you and everything. And I said, oh my God, that's so cool. It has little baby hydras and their little planula larvae, and they love their little mommy hydra. They're running all. Hydra don't have a planula larva at all. So I was wrong, obviously. They weren't planula larvae. Instead, they were this. This is one cell. This is a few hundred cells at least. One-celled protozoan. It's a ciliate related to things like paramecium. It happens to be commensal with Hydra. It loves Hydra. Hydra is its best little buddy. And that's what was in there. I was hoping I'd see some of them in yours. I didn't see any in the ones that we got this time. But they just love it and rub up and down it and rub all over it and zoom all around it and everything. And you see them under a dissecting scope. It's called Corona polyprum. So see, every semester I do this, I learn something. So as soon as I told the person that, oh, that's so cool that they're all the cute little planula larvae playing with their mom and everything, then I go look it up and it says, Hydra doesn't have planula larvae, so I'm like, okay, well, so that can't be what they are, right? There is no planula larval stage, but most cnidarians do go through this type of a larval stage, just not Hydra. So if you ever see cute little things that you mistake for being planula larvae with Hydra, they're actually probably this little commensal cilia that loves it. Okay, so let's try a couple questions. that we're talking about. So which of these animals lacks true tissue? A jellyfish, a sponge, a hydra, or all animals have true tissue? That's okay, did everybody say one's going twice? Okay. So sponges, right, don't have true tissues. How about muscle cells? Y'all remember muscle cells? Can only contract, can affect fluid movement, independent of a rigid structure. Can only pull, never push, or all of those. It's true for muscles. Y'all remember muscle?
Because it only pushes. Muscle only pushes. It never pulls. It doesn't pull. It only contracts. Contraction is pulling. I don't know what B means.
You don't know what it means?
Can-
It means that it
I
can
think-
affect, do you know what a fluid movement is?
No.
Like this would be a fluid movement, right? You just sit there and go, that's not a fluid movement.
We didn't really understand.
Can affect fluid movement independent of a rigid structure. Usually more words mean that it's right. I don't know if it's the case.
Robot.
Yeah, except, see, I wrote these questions, so I don't do it probably the way normal people would question. That's why most of them don't make any sense. Okay, one more, is there one more, then I'm gonna, I actually am gonna finish about the same spot with you guys I did with the other one. I thought I was going faster, but I'm really not. Okay, is there one more? Here we go, we're gonna end it. We're gonna end it, check your numbers, everybody. Joel, your numbers. He's the only person who I know who's not. Seriously, he is actually. He's the only person I know.
It's still like going in. That's what I thought he was talking about. Why is it not going in? Good talk.
Okay, here we go. Why do I feel like this is not going to be good?
What?
Okay, muscles can only contract. What does that mean? Can't muscles relax? Okay, can affect fluid movement independent of a rigid structure. No, muscles need something rigid that they work against, right? Either a skeletal system or a hydrostatic skeleton or something. You can't affect fluid movement without a skeletal system, but they can only pull, they never push. There's no force in relaxing, right? So there's no pushing.
Oh, they
right?
never.
The only force comes through contraction, so they can only pull, they can't push. Yikes. Okay, oh goodness. I said this, did you catch it? The potential sister group or taxon to the clade containing all the rest of the eumetazoans is what? Acial flatworms, sponges, quinoflagellates, or placozoans?
I think I put it in the right one. I kind of did it.
I mentioned a couple different things, right, and said that they were potential sister groups or important groups. So which one was the potential sister group for eumetazoans? Basically all of these tissues, right? Not animals without
Okay. Did we study that one?
Sure.
We did? Black zones?
You guys seem to remember this a little better than yesterday. Maybe that's because it was the day after spring break. You guys seem a little bit more like you're kind of remembering. Okay, are we there? 2 more?
Well, hold on.
Then we finish the last part.
It keeps like switching the numbers. I'm sorry.
Okay, are we there now? One more?
Third time's trying, there you go.
There's still one more though. Check your number. Joel's in there. Thank you. Okay, anybody else? Okay, that's why I finished the last one. Got 4 minutes. Yeah? No? Okay. So Blackozones, right? ACLs are the sister group to Bilaterians. Sponges are not monophyletic. They can't be a sister to nothing, right? And guanoflagellates are much to the outside of this. They're to the outside of sponges, right? Eumetozones is to the inside of sponges.
What was the C?
Blackozones. Huh?
Wasn't the C word a sister group or something?
No.
No?
ACLs or placozones are the two that I gave you as potential sisters.
Wasn't the channel flagellates a sister group?
Guanoflagellates are, I'm not sure guanoflagellates are monophyletic, to be perfectly honest. But they would be potentially the sister group to all animals, including sponges, if you're going to consider them. They were shown as monophyletic in that treaty. Okay, so Cnidarians is a phylum, so we're going to divide it into classes. Hydrozoa, Scyphozoa, Cubozoa, and Anthozoa. You already looked at these 3 in the lab. We didn't really look at these. Hydrozoans are going to be things like Hydra, Obelia, Physalia, the Portuguese man of war, right? Few of the things they call corals. Scyphozoans are going to be jellyfish and things that are like jellyfish, sea nettles. Cubozoas are box jellies and sea wasps. And Anthozoa, the flower element animals, are going to be sea anemones and corals, right? You'll notice that the things we call true corals are all marine. Most of these are marine, but there are a few freshwater things. Hydra's freshwater. There's some little freshwater corals and stuff that are considered hydrozoans. So for a long time, people wanted to know what was the relationship between the different classes of Cnidarians. And there was this debate as to whether it looked something like this or something like this. This one sort of said, you know, the ancestral condition for a Cnidarian is to have both a polyp and a medusa stage, and that the anthozoans, the corals and sea anemones, lost the medusa stage completely. The competing hypothesis said that the ancestral condition is to have only a polyp, and that the medusa was acquired in these 3 classes that have it. So one has anthozoans as sort of the basal Cnidarians, and the other one has hydrozoans. It ends up that the most accepted phylogeny is actually this one right now. And there's a very interesting piece of evidence to support this, so I'm going to point it out. If I was to look at your mitochondrial DNA, what would it look like, you know? Your mitochondria, you realize, are all phylogenetically what? Proteobacteria, right? What does bacterial DNA look like? It's circular. So your mitochondrial DNA is circular. You realize that. Your nuclear DNA is linear, but your mitochondrial DNA is all circular. If you looked at plant mitochondrial DNA, it's circular. If you looked at fungal mitochondrial DNA, it's circular. If you looked at any kind of protist mitochondrial DNA, it's circular. If you looked at a sea anemone or coral's mitochondrial DNA, it's circular. Everybody's mitochondrial DNA is circular, but not these guys. Hydrozoans, cubozoans, and scyphozoans have linear mitochondrial DNA. That's crazy. That's insanely weird. No other animal has that, to my knowledge. No other eukaryote has that. That really argues strongly that this is the right tree, and that this switch to linear mitochondrial DNA happened right here, and that they've all inherited that very, very unusual trait from that common ancestor that they share. So this is now the more accepted tree. So let's just look really quickly. Hydrozoans would be things like Obelia. This is the one that you looked at with the little feeding polyps and reproductive polyps. This is an adult Obelia, goes through Medusa stage. Physalia, the Portuguese man of war. Hydra with this little bud. This is Bolella bolella. It's got this little sail on it. It's a little colonial hydrozoan. These wash up by the bazillions in like San Francisco, where I'm from. This shows you the life history of Obelia. You'll notice that it starts as a zygote, makes a little blastula, undergoes gastrulation at some point, develops into this little planula larva, has the polyp juvenile stages, only one adult. This is not an alternation of generations, right? There's only one adult here. The adult does meiosis to make gametes. Those are the only haploid cells in life history. They fuse to make a diploid zygote. It goes right back to being diploid again, right? It's an animal life history. Scyphozoans are going to be things like jellyfish. Notice that most jellyfish go through a what? Planula larva. They make these little things. It's called actually a strobulating scyphostoma, right? Strobulating because it looks like a little strobolus or a cone. These are the polyps. They bud off into these little things called a phyra, which grow up into adult medusae, right? So it spends a portion of its life as a polyp, but not a very long one. And the prolonged part of the life history in a scyphozoan is actually the medusa. Cubozoans, some of the most deadly cnidarians are these. These are box jellies, like they have these signs in Australia. You hear somebody getting stung by jellyfish and they die like instantly in Australia. It's usually a cubozoan. And then the anthozoans, the flower animals. So these are going to be like anemones and corals. And of course, corals are extremely important because they build
Reefs?
coral reefs, right? So if I show you this is a picture of Australia. I think in the other class they were looking for an animal there. This is Australia. This is Queensland, right? This right here is supposed to be the only living structure you can see from outer space. This is the Great Barrier Reef, the largest barrier reef in the world. The second largest barrier reef is off the coast of Belize, and the third largest barrier reef in the entire world is off the coast of Florida. It's off the Florida Keys. This is me swimming with a giant sea wrasse. Where I think, wow, he studies mushrooms. What could he possibly know about the world? To Cairns, Australia. This is where the International Mycological Congress was in 2006. I got to go swimming off that little pontoon and snorkeling on the Great Barrier Reef, which is unlike any other place you could ever go snorkeling. So corals are incredibly important for ecological reasons because they build these massive structures that house incredible engines of biodiversity in marine systems, right?
Oh, boy.
The equivalent of rainforests in terrestrial systems from a biodiversity point of view. So jellyfish are what? Scyphozoans, anthozoans, or hydrozoans? I think I got one more question, then I'll have you in the same spot where I had the other class. So next time I'm going to talk a little bit, a couple movies, and then try to get you to analyze the balls. That's what I'm going to try to do on Thursday so I can get you up to where we are in the lab. Somehow this week the lab and the lecture is going to come together. Okay, are there four more? That's easy, right? I just said it. What are jellyfish? Are they scyphozoans, ctenophores, anthozoans, or hydrozoans? Two more. Two more, ready? Then we're going to start lab at about 10 after 7, right? So that I have to get that. Is there one more?
Yeah.
I think I got one more question after this. Okay, here we go. So you all put scyphozoans, right? Ctenophores are cone jellies. They're in a different phylum. How about this? The hydrozoan Obelia has a plant life history alternating between a polyp phase and medusa phase. Please get this right. Obelia, that cute little colonial hydrozoan that I just showed you, it's saying here that it has a plant life history, right? That it alternates between a polyp and a medusa, kind of like a plant alternates between a gametophyte and a sporophyte. Is that true? You realize this sentence was in a book they were using for this class before it came. It was published in 2000. That's the way they answer. Okay, everybody there? Two more?
Almost. Take me a while to answer.
Okay, it looks like you're all there, right? 17? Everybody there? Okay, so which animals have a plant life history? No, no.